All right, good morning. That took a ton of cognitive reserve to get that up there. I can't thank Dan enough for inviting me to participate. My very first Friday Harbor was the cognitive reserve one. I learned tons. Some of the ways in which we've put variables into the Harvard Aging Brain Study was based on the inspiration of that conference. So I'm really thrilled to be back and to share with you some of the work that we've done. I am not a statistician. I will put that up front. That's my disclosure. But I play one on TV. No, I play one in our lab. But anyway, today I want to really talk a little bit about impact of cognitive reserve in preclinical Alzheimer's disease. These are the NIH disclosures. These are my disclosures. I do some consulting, but it has nothing to do with the work I'm going to talk about today. Whoops. All right. You know, I like to show this slide because brain pathology and Alzheimer's disease is no respecter of persons. So even though we know that high education and high occupational attainment can be protective in life, we also know that these brain changes of amyloid beta protein and tau neurofibrillary tangles are occurring 10 to 15, maybe 20 years before someone gets a diagnosis of dementia. And I started putting this slide together maybe 20 years ago, and it's updated itself ever since. And we can see that this brain pathology is no respecter of persons. What's important, though, is for us to try to understand what are reserve variables. I love what Dan said earlier about what protections can we really inform patients about so that we can live full, productive lives for as long as we can. This is the Jakob Stern cartoon that we all know about, and I think my goal is trying to understand when does this inflection point occur. And I would love to know it on an individual basis, which is one of the reasons why I signed up for the longitudinal work group, because when will somebody decline? I'm a clinician, so I have evaluated patients, and I have seen this inflection point in people who are high functioning and how they decline rapidly within the space of a year or two to even moderate to severe states of dementia. So what is amazing is that people can maintain function for quite a long period of time, but then this point occurs. So I'm really trying to understand that. But one of the very first references, although Katzman gets quoted in 1974 as being the originator of the term reserve, I actually found this paper from 1968. And this was a pathology paper by Blessed, Tomlinson, and Roth. I still use the Blessed Dementia Rating Scale clinically, believe it or not. And they were the first to notice that there were these people who had high scores, but actually had a low plaque count. And so they began to hypothesize that there would appear that a certain amount of the change estimated by plaque count may be accommodated within the reserve capacity of the cerebrum without causing manifest intellectual impairment. And if anybody has an even earlier reference, I'd love to know, but I think they are the very first persons to coin this term reserve. One of the things that I'm going to be talking about today is the role that reserve may play within this preclinical stage of disease. As I said earlier, the changes in the brain are occurring 15 to 20 years before the clinical manifestations of MCI or dementia. As you go up, you see that there's this long preclinical period. Now, where is that inflection point where it deviates from normal performance and begins to fall down the trajectory of AD? And some of this work was actually inspired not only by the pathology that Laura showed from the Price paper from WashU, but then we had the ability to in vivo see amyloid and plaque burden in living individuals. And this is work from the Harvard Aging Brain Study that Keith Johnson and I did many years ago, in which we found that when you have amyloid pathology, and I'll go through this for the non-clinicians or people who don't live in this world forever, that you see that this is Pittsburgh compound B, that the tracer to amyloid actually flows out of the brain and leaves this blue area. So unlike a PET scan that has glucose, blue is good. And so you want to see this blue in cognitively normal individuals. These are people that we call amyloid negative or A-beta negative. And then when you see patients with Alzheimer's disease, you see a lot of the yellows and oranges and pinks, which indicate a high level of amyloid burden throughout the brain. But then we began to notice, similar to Price and Morris, that there were a whole group of individuals who were cognitively normal, and this is in life, who have a burden of amyloid within the brain that is consistent with patients who are diagnosed with MCI or with Alzheimer's disease. And one of my questions early on was, how can you walk around with a head full of amyloid and still perform normally? You know, what is this reserve or resilience? And so that began some of my pursuit using biomarker evidence. But one of the other things that came about in 2013 when Keith Johnson performed the very first tau scan in a living human being, and this was done at Mass General. And now in the Harvard Aging Brain Study, we have about 130 to 140 individuals who now have had both amyloid and tau scans, PET scans. And one of the things that, again, we notice is that there is a tau tracer here in the entorhinal cortex in clinically normal individuals. We're still trying to understand this because if you read the papers of Brock and Brock, they begin to notice tau in the entorhinal cortex even by the age of 8. So we begin to think, well, is tau normal, and is this part of the normal aging process so that some of the cognitive declines or heterogeneity that we see in aging, is this due to the increase of of tau in the entorhinal cortex? However, one of the other interesting things is that tau in individuals, once it begins to migrate into neocortical regions and outside of the entorhinal cortex, and as amyloid begins to increase in the brain, we begin to think that that is more proximal to the cognitive decline that we actually see in preclinical stages of Alzheimer's disease. And concordant with the pathology studies that we've known all along, it is the tangles or the tau pathology that is more proximal to the dementia. But these are now, we actually see it in clinically normal older individuals. And this has led, of course, to this whole idea of preclinical Alzheimer's disease. In the past, preclinical was always referred to animal studies. And now we're actually using the term preclinical to talk about this stage where people are still cognitively and functionally normal, but showing these biomarker changes. And Risa published this seminal paper on criteria for preclinical Alzheimer's disease. This criteria is only meant to be research-related, and there are various different stages. So there are some people, of course, who are older who have absolutely no amyloid at all in the brain, and these people are, I think, are your cognitive, not resilient, but cognitive or brain reserve. Then there are people who have amyloid alone in the brain, and I'll show you a little bit of data from our group and many groups of what that actually means to cognition. But then people who enter into stage 2 are the people who begin to not only have amyloidosis, but they also begin to show neurodegeneration, and we're beginning to think that those are also the people who begin to show the tau pathology, and of course it begins to have changes on cognition as well. So that's the definition of preclinical Alzheimer's disease that I'm going to be using today. And so some of the work, not only in our lab, but in many labs across the world. This is by Beth Mormino, who has been to Friday Harbor, one of our rising stars, who's now over at Stanford. She published this paper a while ago, which actually divided people into stage 0, stage 1, stage 2, and then there were the group of people that they call SNAP, which is suspected neurodegeneration, but not Alzheimer pathology. And what happens to these individuals over time? And what you find is that on a cognitive composite. So what you find is that people who have absolutely no amyloid or aren't called SNAP, so they're your normal individuals, they actually get better over time, so that there is some practice effect in our longitudinal cognitive measures. I hope that's one thing that we can begin to address when we talk about reserve, because are some of the things that we're really measuring, is it really reserve, or is it practice, or is it some habituation? There are many factors that go into it. What's interesting, though, is that if you have amyloidosis alone, while you're not getting as good, you don't benefit from practice in the same way, what you see is that they're also getting better. So amyloid alone, while I don't think is benign, it's not critical to the cognitive decline that we actually see. There's some interaction that's beginning to occur that actually produces this step 2. Is it the interaction with tau? We've looked at vascular risk. Is there a combination of vascular risk associated with that contributes to this decline? Is it neuroinflammation, of which I think is also a critical player, but we don't have in vivo biomarkers right now to be able to test that hypothesis. But what we do know is that once you enter stage 2, where you have amyloidosis plus neurodegeneration or potentially tau pathology, you will decline over time. What is also interesting, and this has been shown both in our group and in the ABLE group, is that if you have tau alone, you actually never start out doing good, but you're not really declining over time either. And I think that Victor Villamay and Sam Burnham have shown in some of their papers looking at SNAP is that these people actually have some stability over time. Now, this is all hypothetical because who knows what's really going on in the brain in the people who have this tau pathology. Is this the right kind of tau pathology that may be involved in other neurodegenerative diseases? There's still a lot to learn. And this is looking at the primary Alzheimer cognitive composite. In the A4 study, which is a prevention trial using a monoclonal antibody against amyloid in people who are in these preclinical stages. So these are people who are clinically normal, their CDRs of 0, but on a PET scan, they actually show a burden of amyloid in the brain. Now, in the Harvard Aging Brain Study, in order to test whether or not the PAC would be useful in a clinical trial, we began to see how does this cognitive composite track over time in people who have amyloid burden. And what you see, again, is that the people who are amyloid positive decline. They decline in many of the cognitive measures. This cognitive composite has both executive processing speed and episodic memory. It's kind of loaded in that direction. But then you see there are some measures, even in amyloid positive, such as logical memory. In this case, it's the Anna Thompson story. Everybody knows about Anna Thompson from South Boston who gets robbed, and they get that same story over and over again. Well, you can see that people with amyloid burden, amazingly, begin to not remember as many elements as they did, but they actually show a practice effect as well. So again, amyloid burden and whatever is also contributing to this decline is not a benign process. So what about reserve and resilience in relationship to this information? I was blessed to be at Friday Harbor in 2009. These were a special issue of GIN. Some of these papers were already talked about, except that Rich Jones wrote a wonderful paper on conceptual and measurement challenges when talking about reserve. Jakob Stern commented on it as well. So this was actually in 2009 when our ideas of measuring reserve were actually in its infancy. We never shared slides, so I didn't know there was going to be a lot of talk about this residual method, which I really loved, by Bruce Reed and Dan Mungus. And then the work group that I participated in was with Angela Jefferson. And again, it was looking at all of these proxies of reserve. And despite the fact that we had such rich proxies of reserve in the Rush database, she also found it was the word reading list that seemed to confer the ability to maintain performance over time. So in the Harvard Aging Brain Study, I decided when I was designing it that I wanted to throw in measures of cognitive reserve. And so this is work that Rachel did just this past year. She presented this work at AAIC, and she took all of, we did the Hollingshead, but we separated out education from occupational attainment. We had the cognitive activity scale. We had both early life and 40 years to late life. We had education. We had the Amnard IQ, which is a verbal reading list. And you can see that they're like modestly correlated with each other, which gave Rachel some confidence that, well, let's put it into a cognitive reserve composite and see how it holds up. And she was actually looking at functional connectivity. So instead of looking at brain pathology in the way we've been talking about, she wanted to see, let's look at cognitive or functional activity as it relates to cognitive reserve. And so in our sample, we had 243 overall individuals. 182 at that time were amyloid negative. The 61 are amyloid positive. This is an ongoing study. These numbers change as we speak on a daily basis. But what was interesting is that when you divide the group into amyloid negative and amyloid positive individuals, you know, their education, their occupational attainment, their cognitive activities, and their Amnard IQ are pretty equivalent and not different from each other. We have a pretty homogeneous sample. They're all pretty high-functioning individuals, so that doesn't really surprise me. We worked very hard, though, at trying to increase diversity and heterogeneity in that sample. We now have about 61 individuals who are now African American. We now span an age range from like 8 years to 20 years. But unlike the YCAP, which has very rich data of a lot of heterogeneity, we're not anywhere near that. So use that as a caveat. And so then she looked at functional activity and found that greater cognitive reserve on this composite actually showed some significance to functional connectivity, particularly in the frontoparietal network, which on the right side, which seems to show the greatest, but also on the left. You can see a little bit of change in the default mode network, and that in essence, but not in dorsal attention or in the motor, of course, but everything else seems to be somehow associated, indicating that the greater functional activity you have, the greater it's associated with greater reserve. There's no surprise there. I think we would kind of expect that. And then she looked at functional connectivity as it relates to cognitive performance on the PAC. And again, what you see are significant relationships so that the less robust your connectivity is, the greater decline that you see across these various different networks. And then she wanted to tease out this cognitive reserve composite to see what is having the most effect. And what, again, we see is that it's the Amnard verbal IQ that seems to be the most salient. There's some hit, of course, in education, but in our group, it's really interesting because our group has always been somewhat biased in the sense that women did not always have the same advantages of education as men. And it's one of the main reasons in much of the work that I've done is that I've always looked at ability level because based on my own personal experience, my mom graduated 8th grade. She never got any farther than that. And as a kid, I always thought she was very smart. But it made me think that going back and looking at our data, that there were many very smart women in our group who just didn't have the same advantages of education. So it doesn't surprise me we actually see this here. We didn't get the same thing in cognitive activities and absolutely nothing in occupational attainment. And then when you looked at the reserve composite in relationship to the verbal IQ, you can see that the verbal IQ seems to separate the groups a little bit better than this actual reserve composite. So one of the challenges I think that we're going to be facing in our work groups is what is the best way to measure this proxy. Maybe Dan is right. We should get rid of this whole idea of proxies and just really look at cognitive performance. I think that longitudinal data over time will give us more insights. But right now, in our sample anyway, it looks like the Amnard verbal IQ gives us the best separation of the groups. So what about that in relationship to preclinical AD? So I published this paper back in 2010 in Annals of Neurology, and this was across, now you remember this is very early days of PIP-PET imaging, so we only had 66 normal controls, we had 18 individuals with MCI and AD, so we looked at it across the disease spectrum. And what we found is that the people with high IQ actually performed consistently on a cognitive composite. Now, one of the technical things we did here, and this was inspired by an article written by Gene Alexander, and I think it was in 1997, when he looked at cognitive reserve. And so what we did is we residualized MMSE on Amnard in order to control for the effect that dementia might have on your word list reading test. So this is actually a residualized effect of cognitive reserve. What we found, though, and I was, at the time surprised, that when you only looked in the amyloid positive individuals, all the lines went down. And it indicated to me that somewhere in that inflection point, cognitive reserve loses its protective effect. But I have no idea where. This would be great when we have longitudinal data to figure out where is that inflection point. People who are working later into their older years need to know, when should I retire? When am I not going to be as competent? And I think it would be really great to have some idea of that. And when I only looked in the normals, what was interesting is that while I got a effect of Amnard on reserve, I did a reserve on performance, I lost the interaction effect with amyloid burden. And it was Keith Johnson who said to me, well, maybe our tests just aren't challenging enough. That this cognitive composite is actually not strong enough to be able to see this impact of amyloid burden in normals. So we partnered with Herman Buschke. We did the memory capacity test. I brought people back in January, to do this test. And once again, we found that on challenging measures, people actually who were highly educated or high ability level were actually able to maintain performance in contrast to the people who had middle or low reserve. So that actually began the the pursuit of different, more challenging tests to look at preclinical AD in normal individuals. And then just recently in 2017, with the help of Kate Papp and Beth Mormino, I wanted to look at this in relationship to tau burden, now that we had amyloid and tau together. So again, we had 133 normals, and we had 17 MCIs and 6 ADs. We tried to replicate what we did in the Annals paper across the disease spectrum. Instead of doing a residualized effect of AMNART on MMSE, what we ended up here is creating a variable on on clinical status, and that was one of the moderating variables that was thrown in here. And so in a typical David Bennett fashion, we created these models. One, we wanted to look, what's the impact of amyloid alone on cognition in relationship to reserve, cognition in relationship to amyloid alone, in relationship to tau alone, tau PET imaging. And then if you put both in the model, what happens? And then to look at amyloid and then the interaction of amyloid plus cognitive reserve, and then to look at tau and tau plus cognitive reserve. And so what we found is that when you have amyloid in the model, and you can see we used age, education, VIQ, clinical status, and amyloid, that amyloid is related to cognition. And the greater the amyloid, the worse your cognition. So there was a negative value here. When we looked at tau alone, you can see that it had an even more powerful effect than amyloid alone and was also clinically significant. But what was really interesting here is that when you put both in the model, typical to all the pathology studies, is that amyloid drops out and tau is the thing that seems to have the greatest impact on cognition. And then if you look at the interaction of amyloid, you can see that there's a trend to amyloid and cognitive reserve. But when you look at the tau and cognitive reserve interaction, again, that is highly significant. We then looked at it in normals alone, or these are the graphs. And so again, this is amyloid, and you can see that we have a similar kind of graph that we had in the Annals paper, where the higher your cognition, the more stable your performance. But then here, even though people with high IQ are tending to do a little bit better, you can see that there are declines across the board in these slopes, which may suggest that once you have tau and amyloid in the model, that you're beginning to lose the protective effect of cognitive reserve. We then looked at it in normals alone. We did the same models. And again, we found that there was a significant relationship with amyloid to cognition, an even stronger relationship to tau and cognition, but we lost the interaction. So here, we didn't see any kind of interaction with cognitive reserve. In part, this was because our cognitive variable was the MMSE, and that was because we didn't have a wide range of cognitive variables in the clinical group, the people who had MCI and AD. So I think we found this in relationship to the quality of the tests that we were using, and we began to use it in relationship to the PAC, and we found that there was an interaction. But we didn't publish it, because we wanted to go back to the drawing board and to look now longitudinally, because this was cross-sectional data. What happens over time to cognitive reserve? Rachel has been working with me on this. It's a slow process of trying to build the data up. And what we found, again, is that over time, you can see that if you look at this interaction of amyloid, VIQ, and time, that we see a significant relationship where people who are more, who have higher ability level continue to do better than people with lower ability level. But when you look at it in relationship to tau, even though this is significant, this was not significant, this is a trend. When you look at the tau relationship, you can see even though there is a significant relationship, that the lines are really going down. And again, indicating to me that while you have reserve, and while you're performing a little bit better than the people with low ability, you're not doing as well. So we began to look again longitudinally, and this is work that Rachel did. So now looking at PAC performance in relationship to VIQ, what you see is that the people who are who have IQs of 130 or 122 or 114, that they're all separated, they don't perform as well to each other, but this was not clinically significant. I'm showing this data because this is a work in progress. It's not fully cooked yet. But when you put amyloid, when you divide the groups up between amyloid positive and amyloid negative, and you look at the relationship of inferior tau to cognitive change, what you see is that the people who are amyloid negative have this is non-significant data. People who have higher ability do a little bit better than the people with lower ability, even in the face of inferior tau. But when you put amyloid positivity in the model, when you look at those people alone, what you see is that these people with high reserve are actually continuing to decline. Even the people with low reserve are actually beginning to decline. Again, like I said, this is a work in progress. I don't make anything of this at this point, but I wanted you to see some of our work, help us to think this through. I know Rachel's going to have a million questions for everybody, but we're going to wait and try to collect more data to see what the real story is. So in summary, we see that longitudinal analysis show that greater A beta and inferior temporal tau deposition is associated with worse cognitive performance, that cognitive reserve tends to modify that relationship. We think that inferior temporal tau had a stronger, more direct association with poor cognitive performance than A beta burden, and that cognitive reserve may exert an initial beneficial effect by reducing detrimental consequences of A beta and tau burden, but I think it loses its protective effect over time. And in the remaining couple of minutes, I want to say why this is important. And some of you are not clinicians. I'm a clinician. I get a chance to observe the impact of reserve in people. And I initially, back in 2005, decided to develop an IQ-adjusted method so that I could clinically look at people with high ability level and determine whether or not I can detect who's at risk for further future cognitive decline. And just to illustrate, the average range is at the 50th percentile. As neuropsychologists, we tend to measure 2 standard deviations below the mean. And so that leaves you with an impairment range at the 2nd percentile. But let's say you live up here at 130, and everybody in this room probably qualifies for that. If I was to use the same standard for the general population at the 2nd percentile, you would have to drop 4 standard deviations before I would call you impaired. And that just didn't seem right to me. Why would somebody, the normal Joe, only have to drop 2, but you got to drop 4 before I would indicate that you were at risk for something? So if you do an IQ-adjusted method, maybe your cutoff score is at the 50th percentile. Now, that seems a little bizarre for neuropsychologists or even statisticians to think that less than the 50th percentile, if you have an IQ of 130, would be impaired. But let me show you some results. So I did this study, and I scored everybody according to the standard literature, and so everybody scored in the normal range. But when I applied IQ-adjusted methods, I discovered that there was this subgroup of people who actually score lower. And so I targeted these people to see what happens to them over time. And what we found is that these people who had IQ-adjusted memory impairments actually, over 3 years, were 11.2 times greater for going on to develop MCI than the people who were normal. And again, you can see in here some of these, trust me, maybe you can't read them all, but you're dropping, your change is really occurring in tests of memory, in tests of naming, and also in semantic fluency. So I saw this gentleman, 72-year-old physician. He's an entrepreneur. He has an IQ of 130. And he was complaining a little bit about his memory. And this was in 2009, at the time of our Cognitive Reserve Friday Harbor Conference. And what I found in him is that he had little dips for his ability in semantic fluency and delayed recall, and also in multiple choice and in naming. But I didn't have any guts, and I didn't have a drug that I could put him on to be able to say we should target him, so we waited. So then in 2010, now his MMSE is down to 27. His categories dropped from 36 to 32. And he's fluctuating in his memory performance here. But on the free and acute selective reminding test, which I think is quite sensitive to temporal limbic amnesia, he's beginning to show declines. And he has a little bit of a decline in his naming. And then by 2012, he has the same MMSE score, but now he's at 0 in delayed recall on a word list. He's got an 8 out of 10 in multiple choice. And now look at his free and cued. His naming is at the first percentile. And the last time I saw him, he actually had Alzheimer's disease. We put him into a clinical trial, and it was too late. He went into the clinical trial at the stage of MCI, and we were not able to reverse his course. Part of the reason I'm saying that is that eventually we are going to have a drug. And the trick is going to be identifying people who have reserve that we don't miss so that they can have the opportunities to get a disease-modifying drug before it's too late. If we wait too late, then it's not going to be useful. So it's this whole idea of identifying people who have high reserve and being able to target them for disease-modifying therapies. And this was his graphic. You can see that he still had very good executive function even up until 2015, but it was in the areas of language and semantic processing along with memory that he actually showed decline. So that's all I have to say. I want to thank you. I don't do this work by myself. It's a whole group of individuals, and I'm inspired every day by all of them. And I'm just thrilled that we're going to be tackling this whole concept because it means something clinically to me as well. So thank you very much.